Please raise your hand if you've ever had braces, aligners, or any orthodontic treatment. Now keep your hand raised if it led to jaw pain, jaw clicking, or any oral discomfort. See, I hypothesize that this is related to the maladaptation of mechanosensitive tissues surrounding teeth. We can see here that as this little rodent bites down on food, mechanical forces of tension and compression traverse the tissues that surround its teeth. Now, at the UCSF School of Dentistry, we are particularly interested in optimizing clinical interventions such as dental implants and orthodontic treatment. But the problem is that we do not know how much force should be applied so that these treatments are still effective but do not exceed a threshold past which tissues are damaged. This is where my research comes in. An important tissue in this system is the periodontal ligament, or the PDL. As shown in the figure, the PDL is a rubber band-like tissue that connects the root of a tooth to surrounding bone. Now, this keeps our teeth in place as we chew. Fibroblasts are the primary cell type of ligament, so I used a mouse fibroblast cell line to mimic human cells of the PDL. Then, to mimic conditions of tension and compression, I manipulated mechanosensory ion channels TRPV4 and PIEZO1. These ion channels are found on the cell membrane and open and close under mechanical loads. TRPV4 is activated by tension, while PIEZO1 is activated by compression. I then treated the cells using synthetic molecules that either activate or dampen activity of these specific ion channels. I estimated changes in cell activity by staining the cells using a fluorescent dye to visualize mitochondrial activity as shown in red. After measuring and quantifying these fluorescent signals using a computer program created by our lab, I obtained the following results. After adding the dampener molecules, mitochondrial activity was dampened as we expected. However, after adding the activator molecules, mitochondrial activity increased for PIEZO1, but intriguingly not for TRPV4. This gave us an insight into the potential importance of physical tissue deformation in activating the TRPV4 ion channel. In other words, it appears that we cannot manipulate tension using synthetic molecules alone, but we are able to manipulate compression using these molecules. To apply these results, our lab is putting these molecules into rodent food to identify optimal thresholds for mechanical load in vivo to continue improving clinical interventions for humans. My name is Tiffany, and thank you for your time.